Hi, my name is Tim Storr and I'm a professor of chemistry at Simon Fraser University in Canada. This video is a complement to a recent JBIC review article titled A Role for Bioinorganic Chemistry in the Reactivation of Mutant p53 in Cancer. So my group's interest in protein misfolding really started in Alzheimer's disease, in particular the extracellular plaques that develop in the brain. These plaques, the major constituents is the amyloid beta peptide, as well as a high concentration of metal ions, iron, copper, and zinc. And it's particularly the redox activation of copper and iron, redox cycling, and generation of reactive oxygen species that got us interested in this area of bioinorganic chemistry. So the p53 protein is fascinating. It has been coined the guardian of the genome and plays a critical role in the cell, sensing DNA damage and either shutting down the cell or instigating repair mechanisms. However, in over 50% of cancers, the p53 protein is mutated and doesn't act in accordance to what it is supposed to do. And so we've developed small molecule compounds, many others have as well, to try and reactivate mutant p53. And an additional point that really piqued our interest was the fact that p53 mutations lead to aggregation and the formation of amyloid aggregates. And so this comes back directly to what we were doing with our Alzheimer's disease research.
So I've always been interested in the fields of medicine and chemistry, and on a personal level was motivated to do research in this field when a close family friend passed away from cancer. And p53 is such an interesting protein that's really at the center of many cellular processes, and it has a large impact in cancer development. And so I think this project was really the perfect protein to bridge the fields of medicine and synthetic chemistry. For my PhD research, we aim to combine two strategies for restoring p53 function as a therapeutic strategy. By designing multifunctional molecules that can target both mutant-induced cavities as well as restore zinc binding to the protein, we can design effective molecules that can both prevent p53 aggregation as well as restoring wild-type function to the mutant proteins. One of the things I enjoyed most about this research project is the collaborations that I've made along the way. In particular, with Christian Guédon, we met at a research conference in Hawaii and started a collaboration that involved a 3-month stay at his lab in Strasbourg, France. And I learned so much from Christian and his lab in the field of molecular biology, and really credit a lot of my success to have meeting Christian as well as my supervisor, Tim Storr.
All right, this has been a fantastic interdisciplinary project so far. It's been a real pleasure working with graduate students Jessica Miller and Calvin Kwan, as well as our external collaborator in Strasbourg, Dr. Christian Guédon. And again, I think there's a really neat role here for modulating intracellular zinc concentrations to remetallate mutant p53 and reactivate this important protein in terms of moving forward with cancer